So I took off a part of the title. Actually forgot that I was going to emphasize the motor recordings and the voice recordings. I'll tell you a bit about that. The thing is that we're in the midst of starting to analyze that kind of data, so I have less meat to show you there. But I wanted to give you a more, in preparation for the discussion that we already started, in thinking about the challenges of how you might use neurobiological and also computational tools to make a difference in identifying psychiatric disorders and also in intervening. So in the lab, the things that we do well is we record fMRI and EEG and do a bunch of signal processing and different types of analyses. In the last few years, we've also been getting, I've done motor control for a long time, but we're starting to get more into eye tracking and these kind of full body recordings that you can get with a Kinect camera. These are the wonderful students I work with, and the idea is to use these tools in order to think about how to make a difference in the case of autism and more generally with respect to developmental disorders. So if you look at the definition of autism, which is the main focus of the lab, and I always love this picture of the different DSMs, you've already seen one of them, right? You get a feeling for how this is the first one from, I think, the 50s or 60s. You can get a feeling for how they've changed over time, and that's true for all of the, definitely for the developmental disorders. Here you can see the change that has occurred in moving from DSM-4 to DSM-5. The criteria have changed. And this is, it's basically a nightmare for a scientist because it means that the population is changing. Even if the changes are slight, it's changing over time. The people who are diagnosed with autism today are not the same as those who were diagnosed 20 or 30 years ago. So, looking at results of studies 20 years ago, you always have to keep that in mind when you interpret them and think about how they relate to yours. So in case you don't know, Asperger and PDD have left the world. They no longer exist, which opens up really interesting identity questions for people who have been diagnosed with Asperger. They have these nice T-shirts that say, I had Asperger's syndrome briefly between 1990 and 2000, something like that. And so now everyone gets a full diagnosis of autism, which can be mild, moderate, or severe. Okay, we'll get back to that in the discussion. Actually, I have a lot to say about the kind of arbitrary decisions that are made here. Another example is that today with the DSM-5, you can have both a diagnosis of autism and ADHD. And according to the DSM-4, it was, what's it called? It ruled out, okay? You could have either, but not both. Okay, so the question that we're interested in is what's happening, right, in the brain, assuming that all of these behavioral problems are emerging because of some changes in the brain. For many years, people have asked the question, where is autism in the brain, which might be a rather naive question. There's been, I won't get into any of these research, there's a long list of implicated brain areas, depending on the particular tasks that individuals with autism are asked to perform in an fMRI setting or an EEG setting. They show different abnormalities and brain responses in a wide variety of areas. And for each one of these areas, there's actually an entire theory about how dysfunction in that particular area might be related to the autistic symptoms. But in my view, I said it's naive because autism is for sure not a lesion. It's, you can't knock someone over the head at the age of 15 or 20 and create autism. That doesn't exist. Okay? It has to be, it's a developmental disorder. The changes have to occur early in development. The question is whether it's already, right, at what stage of early development between the age of 0 and 3, between conception and 3, something like that. So there's a wide variety of scientists who are rather than thinking of it as a brain lesion, have proposed a bunch of theories that are more about widespread abnormalities that likely occur throughout the brain, or at least many parts of the brain, and that have to do with these phrases like excitation-inhibition imbalances. A lot of this comes from genetic work, where there's an association between, for example, GABA receptors, mutations in GABA receptor genes and autism, mutations in structural synaptic proteins like SHANK and Nerexin and others. And so the idea is that the neural network is set up differently. There's also a large comorbidity between autism and epilepsy. About 30%, 20 to 30% of individuals with autism at some point in life will develop epilepsy, as opposed to 1% in the general population. And so that's how a lot of these ideas came about. There's a lot of research about connectivity. And so you end up with this idea that neural processing is altered in widespread areas. Okay? And so one measurable aspect of this, it's a bit difficult to measure excitation inhibition. There are different, so Oren Shriki does nice work about modeling that with avalanches. Another avenue, or perhaps another view of these alterations might be in the reliability of the neural responses, and that's one aspect of the story that I've been captured by. And so that's one of the things that we focus on in the lab. And to explain that, I have this example of a single neuron from a macaque monkey, where the macaque is being presented with exactly the same stimulus over and over. And what you see here is a raster plot. Each line is a trial. You can see the stimulus presentation here at time point 0 and the neural response thereafter. And you can see that even in a healthy control animal, there's very large variability across trials. The neuron, and not just in the timing of the spikes, but also in the overall firing rate. The neuron sometimes fires very strongly, and on other trials, barely responds, okay, like here. And this raises many questions about neural encoding and so forth that we won't get into, but the idea with respect to autism is that perhaps these widespread issues might be related to the reliability of neuronal responses, with this underlying hypothesis that children with noisier or less reliable neurons might have developmental problems. Notice that I'm not limiting myself to autism. I'll talk about the divergence to other disorders as well. So why do I think that there's a relationship? What we do in the lab is we measure the same story, not with electrodes inside the brain, but rather with EEG and fMRI, and these are extremely boring experiments. We typically do it with very simple sensory stimuli, like a checkerboard in an annulus around a fixation spot. We simply show it again and again across multiple trials. And we look at the reliability of, let's say, the visual system to this stimulus across multiple trials. So one way of doing it is focusing in on the visual areas with fMRI and looking at the hemodynamic response. Notice that these are sluggish responses. So hemodynamic responses to a repeating stimuli. And then you can measure trial-by-trial variability in different ways. One simple measure would be to just look at the standard deviation of the peak of the response, and that would give you an estimate of how reliable the visual system was in this particular subject. Okay? You can do the same thing with EEG, where you could utilize the temporal resolution and not only look at the response at an early stimulus-evoked response, the variability during this time point, but also variability in other time points. I forgot to mention that with fMRI we can separate variability of the local responding neural system from variability that's apparent across the entire cortex, that's more widespread and maybe has to do with neuromodulation and lots of other things. So, yeah.
What is the meaning of the fMRI of response after six seconds? Why would you anticipate response after so long time?
Because the fMRI responses are hemodynamic responses. They require the vasculature to open up and then close back. A typical fMRI response takes 6 seconds to reach its peak. So the vasculature opens up slowly and then closes slowly. That's one of the limits of fMRI research, where you have to space your
What was the stimulation for the fMRI that it took 6 seconds?
The stimulation doesn't take 6 seconds. The stimulation takes 2 seconds, okay? And then you wait with a black screen and nothing happens, okay? You wait for the response to evolve, and you quantify it. That's how all of fMRI research works with respect to any stimulus-evoked response you can think of. So we do. So I'll give you an example of a study that looks at the variability of responses in individuals with autism. This is a study that I did at the end of the postdoc with Marlene Berman in Carnegie Mellon, where we invited high-functioning individuals with autism who were older, at the age of 18 to between 18 and 30. And we presented them, we did these boring experiments with stimuli in 3 different domains, in the visual, auditory, and somatosensory domain. This is a manifold. It allows you to control an air pressure system and send out air puffs to the hand in our case. And so what this allows you is to have 3 independent experiments across 3 different systems, sensory systems, and see whether you get a converging answer, okay? And what happens when you compare individuals with and without autism is, in each case here, I'm showing you the response. These are the mean responses. This is the standard deviation across trials. And in each case, it's in a different sensory modality. I didn't want to get into how we localized it. It's very standard. We actually localize it with another run. But what you find is that in individuals, in controlled individuals, and autism individuals, you have very similar response amplitudes, such that the mean peak across trials is of similar amplitude. But you get a significant difference in the trial-by-trial variability with an increase in the variance, in the standard deviation across trials in the autism group. Okay? And once you have your signal amplitude and your trial-by-trial variability, you can compute a signal-to-noise ratio in the engineering sense, okay, which is just the signal divided by the variance. And you can see that there are considerable differences across the groups. They around 25% differences in signal-to-noise ratios. So there have been several follow-up experiments to this. There have been other labs who have shown this already, with both with EEG, and we also did we reproduced this with another group of individuals with autism and found the same thing again. But it's important to you know, we're often biased to look at results in this manner, where we look at the means across the groups, okay? And we tend to avoid the single subject distributions, which are extremely important, particularly when we're thinking about a psychiatric problem. And what I'm plotting here are individual subjects. Okay, you can see that these studies are not large. They're often, so here we had 13 subjects in each group. What you see here is the amount of variance in one sensory modality versus the other one. And you can see that the individuals who have variable responses in one sensory modality also have variable responses in the other sensory modalities, as if this trial-by-trial variability is a personal trait. Okay? And what's particularly interesting are these individuals with autism who are off the scale, who are entirely not overlapping with the control subjects. And this is true for practically every neurological, right? We saw it in the previous lecture with respect to computational modeling and the results there, where you saw that dyslexics, not all the dyslexics had the change in the computation. And this is true also here, and that's something keep in mind when we continue to how we're going to solve autism. So,
do you see the same things in behavior? Behavior is more variable?
I'll show you an example of variable behavior in a second. Yeah.
Regarding the fMRI study, how much sense you're looking at the variance of the hemodynamic?
Can't hear you.
You're looking at the variance of the hemodynamic response. Wouldn't it be more accurate to say the variability is not in the function of neurons, but is in metabolism of astrocytes?
No, because we did a lot of control analyses to make sure that the hemodynamic response itself doesn't differ, not in terms of timing and not in terms of amplitude. We've reproduced the same results with EEG, where you don't have that issue. You have other issues with EEG. But the idea is that you have converging evidence for multiple types of neural measures. And sure, we're not measuring neurons directly here, but this issue of variability is a recurring theme, regardless of how you measure the individuals. Okay. So, so we have a whole theory about why neural variability would be specifically important in autism and in the evolution or the development, not evolution, the emergence of autistic symptoms. The main idea is that we're thinking of an early brain. If the neurons and the neural responses are more variable early in development, that this will have a lot of consequences for how the child perceives the world and how it learns to how different associations are created, and so on and so forth. I don't want to get too much into this. The purpose is to give you an example of a neural measure that shows a difference in autism as part of a larger. I think that this neural measure is particularly important, and we're pursuing to measure it also in toddlers. I'll show you that. So I'll jump around a bit. So we're now measuring similar things with EEG actually in sleeping toddlers. So we've recorded about 10 toddlers with autism and 10 controls in a sleep lab. We're presenting them with auditory responses during sleep. Unfortunately for us, it took us a long time to get this protocol working, so I don't have enough data yet to do a serious comparison. But I wanted to show you these. These are ERP responses across multiple auditory trials during different sleep stages. And you can see that we get similarly robust auditory ERPs during sleep, which will allow us to look at sensory reliability even during different sleep stages. And that's the only way you can look at children in this age group in what I think is a meaningful way. But so that's our extension of the previous fMRI study to looking at younger ages and seeing whether these issues are evident from the get-go. I have a bunch of stuff to show you. So, okay, so this is with respect to neural variability. I'll continue to jump around. This is an example of behavioral variability in autism. This is from voice recordings, a nice study by Yoram Bonnet, where he recorded children reading out single words. So they had a list of 60 words, and what he did is he calculated the fundamental frequency of so the pitch here is the fundamental frequency of their speech. Each point here is a different word. This is a control child, this is an autism child, and what you find in autism is much more variable, much larger pitch ranges and larger variability in the fundamental frequency found right across utterances. There are other ways of looking at speech recordings which are really interesting. There's oh no, I'm blanking out on the name. In any case, okay, so you see here some behavioral evidence that's related. There's also some work with iSaccades showing that the endpoint of the saccades are more variable in children with autism when they're performing saccades to different targets. So, okay, so another two avenues that we're pursuing with respect to the variability is we've just started collecting eye tracking data. This is an eye tracking room that we've built inside the Soroka Hospital. I'll tell you in a moment about this large-scale database that we're building, and this data is part of that large-scale database. So we have children come in as closely as possible after their diagnosis. We tie them into this chair and have them watch different types of videos. This is an example of what you see here are fixations of typical children that we started with. This is my daughter Tamar at the kibbutz. We purposefully use ecological stimuli that have lots of social information, but also lots of alternative things to fixate on, because we're hoping that this will, so this kind of information about the eye fixation tells us not only about ocular motor control. You can look at smooth pursuit movements and saccades and the accuracy and how much jerk there might be there. But it also tells us a lot about social preference, and by looking at pupil diameter, we can also extract changes in neuromodulation, which are one source of variability that we're thinking about. Okay, so that's one avenue of research. Another avenue, which is totally in its infancy, is to record children with a Kinect camera. We're just now starting to look at the kinematics of whole body movements that we're acquiring right now, still from subjects in the lab. But the reason that this stuff is interesting and that I'm telling you about it is, even though these are very premature and initial directions, okay, we're going to jump a little bit, is because this all fits within a larger framework of an effort to understand early autism development that's happening at Ben Gurion. We call this the Autism Forum. We're a group of researchers. So when I arrived at Ben Gurion 4 years ago, the first thing I did was find Gal Meiri, who is a pediatric psychiatrist in Soroka. He does most of the autism diagnoses in Soroka. And we created this forum where we meet every 2 weeks with all the clinicians in the area. This is Chen Steierman from Maccabi Health Services, Analia from the Child Development in Be'er Sheva. Between the three of them, they diagnose something like 400 children with autism, new children, every year in Be'er Sheva. Be'er Sheva is the only place in the Negev where you can get this diagnosis, more or less. And so this is, the fact that Soroka is such a monopoly gives us a great opportunity to do the research inside the hospital and catch a huge number of kids at the earliest stages of development. And one of the big criticisms about autism research is that a lot of what we know, and that's true for my studies as well, is a lot of what we know is about older ages. And that's already 20 or 15 years at least after the initial emergence of the symptoms. So how do you know that what you're measuring is even related to onset? And so there are other scientists. Idan Menashe is an epidemiologist. Chava Golan does molecular biology. Al does genetics. Eitan does computer science. And the idea is to attack the problem from multiple different directions in order to get a large variety of information, okay, which will be incorporated in a database. And so far, we've started from the easier parts, which are collecting the clinical history and all of the behavior. So all of the kids do ADOS assessments. And we're adding the other parts as we go along. We already have, some eye tracking and some EEG. We have a dedicated team that works inside the hospital. So these are university rooms that are right adjacent to the autism clinic, which is very useful, where we do the eye tracking and a lot of the behavioral testing. This is our capture of, in terms of families who have agreed to participate in our study since January 2015. So you can get a feeling for just the numbers in Soroka. Once they sign a consent form, we can dig into all of their records, this is the age range. You can see that most of them are captured before the age of 4, so we're looking at the very early onset of autism. Then we can start digging into their clinical records. We can see a lot of interesting things regarding the population in Be'er Sheva. There's this, when we're talking about autism and the heterogeneity across individuals, there's a lot of heterogeneity within autism. Autism is a kind of a collection of very vaguely described children. About a third of children with autism have regressive autism, which means that they developed pretty much normally until the age of 1.5, until 18 months, and then regressed. Okay, and about a third of the parents report that the children were already speaking, saying a few words, they were socially interacting with the parents and their siblings, and then there's a disappearance of these abilities. Okay? So if you think about the biology that underlies these two parts of the population, my guess is that they're going to be different, that the mechanism that causes autism and an abnormal development from the get-go might be different from what's happening in regressive autism. So that's one example. Another, when we think about the genetics of autism, there's a lot of important information, which is, so Soroka and BGU have made a killing on intrafamilial marriages in the Bedouin community because it's a lot easier to find genetic markers of disorders. So intrafamilial means that both parents are first cousins. And that means that 12.5% of the genetics are the same. Okay, so if you have children of parents who are unaffected, and some of them have autism and others don't, it's a lot easier to identify the genetic cause. And again, the idea is to use these this kind of information to separate the children with autism and see how all of the neurobiological measures that we're measuring and everything else differs across these subgroups, because there's no reason to expect the biology to be the same across the entire population. 93% of the kids who are diagnosed with autism were also born in Soroka, so we can get into their records from the maternity ward. You can see a very preliminary analysis of risk factors based on different anesthesia agents during the when giving birth. Don't take this too seriously because there are lots of controls here that haven't been run, but you see differences in risk depending on what happened, what the mother had during the birth, also what the distress that the offspring had. We collect sensory information, so this is parental report. There's lots of very strong sensory sensitivities in autism, but again, they're not prevalent in the entire group. They characterize a part of the group. Okay, so in thinking about brain development in children who have very severe sensory sensitivities, you might expect that to be different from the 50% of children with autism who have typically distributed sensory sensitivities. The same goes for sleep. The relationship between sleep and sensory sensitivities is a hot topic for us right now. And we just finally got IRB approval to collect genetic data. This only took 1.5 years. And so now we'll probably do this with saliva. It's actually Dan Menashe and Chava Golan who will do all of this. And so you can today get pretty much free genomic sequencing for autism samples from several consortia in the US and Canada. So you don't even need to invest money. So the point of all of this, I'm going to jump back in just one more minute and I'm done. Okay, is this thought that you need to think of autism, and for that that respect of any other psychiatric disorder you can think of, rather than as a spectrum of severities, as a puzzle of different disorders, where with the expectation that each one of these disorders is going to have different biology. And the big question is, do you have 10 disorders here, or 10 different physiological problems, or do you have 300? Okay, and in order to identify these, you need a wide variety of data, not just the neuroimaging pieces and the behavioral pieces, the eye tracking and things that I do, but also the other pieces of the puzzle that are going to come from all of the clinical information, the background of the families, the genetic pieces, the molecular biology pieces. The more pieces you have, the more I think this is going to get interesting in being able to identify a specific subtype of autism with a homogeneous which you could actually characterize as a single disorder. Okay, and the thought is then to to think about how to the million-dollar question is how to change or how to intervene with that particular type of autism. So another part of this research in Soroka is integrating therapies and trying different For example, we have several hypotheses about children with autism who are more likely to be prone to epileptic seizures because they will have, let's say, epileptiform activity in their EEG and will have some genetic polymorphisms and so on. Identifying subgroups of kids like that and trying different anti-epileptic drugs, which people have been discussing for a while now in the field, those are the thoughts that I think that we're going to pursue in the next year or two. And that's it. Thanks. Yeah.
So this high functioning autism is very often associated with what's called the right hemisphere deficiency, which means some sort of problems with integration, gestalt, integration of detail. You didn't say anything about that.
Yeah, I purposefully try to stay away from explanations that are not. So that seems like something that's very difficult to measure in my mind. And particularly difficult to measure in these very early stages of autism. And I'm specifically, the entire focus of the research program is to see what happens during the age between 1 and 4 in order to think of developing the interventions at that, right, with this whole idea that the brain is there's more plasticity here, there's more opportunity to realign development in its proper direction. And it's so it's difficult for me to think about the kind of measures that you would use at these.
Any asymmetry in the development of the right and left hemispheres?
So we did a very big project about anatomy, for example, and we found almost no anatomical differences between the age of 6 and 35 in a database of 1,000 scans. And I'm a bit skeptical about. So it's part of the problem in the diagnosis of autism. There's a wide variety of reports about autism which have been contradicted in many ways, and I think that part of it is because there's such variability in the population. For example, I don't think that everyone with autism has a lateralization issue. I would get, if some of the kids, once we start scanning them with MRI, show a difference in, I don't know, hemispheric volumes or something like that, then that would be really interesting to pursue. Yeah?
It's very interesting, but I'm not sure how to understand. You're talking about larger variations in pitch ranges and neural variability and all kinds of magadim and all the biology. But as a clinician, one of the extreme characteristics that you see in autistic behavior is it's non-variable, repetitive.
So rigidity. But that's typically the rigidity has to do with not wanting change or not being able to cope with change and focusing on a specific repetitive behavior, right? Like flipping a wheel or something like that. One of the thoughts about how repetitive behaviors fits with this story is that if your brain is constantly perceiving the world in an unstable manner, that the repetitive behaviors might be a behavioral compensation for this situation, because the repetitive behaviors create stability. Okay, so they tend to do the same thing over and over and over, be fixated on a single subject, know everything about trains but nothing about other things. So that's my hand-wavy explanation about that.